Hello. Good morning, everyone. Today I will discuss some aspects of vitamin B12, and you will realize that it is an interesting, curious, and intriguing vitamin, and its role in body is being needed. There are four forms of the vitamin B12. We will call B12 as cobalamin, and four variants of the vitamin B12 are cyanocobalamin, hydroxycobalamin, methylcobalamin, and S-deoxyadenosylcobalamin or adenosylcobalamin. Now, among these four forms, natural forms are hydroxy, methyl, and adenosyl, whereas synthetic form is cyanocobalamin. Now, cyanocobalamin and hydroxycobalamin are transport forms, whereas methylcobalamin and adenosylcobalamin are the coenzyme forms, which actually take the role in biological process. All these four varieties of vitamin B12 are in clinical use, and later on we will realize that hydroxycobalamin has a special role in cyanide poisoning and sometimes in ethambutol. Now, sources of vitamin B12 are many. Mostly, the sources are animal sources, but many interesting things will be discussed in this section. Now, basically, vitamin B12 is formed by anaerobic bacteria, Propionibacterium and Lactobacillus, and some legumes also, which are contaminated with soil bacteria, would have small quantities of vitamin B12. In general, it is not found in plant sources. Many vegetarian sources of B12, such as fermented foods, algae, seaweed, spirulina, yeast, and mushrooms, may not contain enough B12 which body can assimilate. Animal stores bioavailable vitamin B12. Particularly, they have a lot of the B12 in their liver, particularly other organs. The more important is liver. In addition, B12 congeners are also found in milk, eggs, mussels. The amount of B12 synthesized by human intestinal flora is very small, negligible, and therefore it is unlikely to contribute to the significant. Vitamin B12 in the form of adenosylcobalamin is a predominant B12 form which is found in meats, while in meats rest forms are hydroxycobalamin and methylcobalamin, whereas methylcobalamin is the predominant form in milk and eggs. It should be understood that cow and buffalo milk contains some very small amounts, that is less than 2 microgram of vitamin B12 per cup of the milk. Therefore, large amount of milk has to be consumed to meet with the daily needs. Now, human milk also contains some amount of vitamin B12, and it has been found that colostrum contains the highest amount, whereas the amount of B12 in human milk depends on the amount of the B12 taken by the mother. Higher the intake, greater is the amount. However, it varies from 590 to 756 nanogram per liter of human milk. Eggs also have some vitamin B12, about 1.6 micrograms in 2 hard-boiled eggs. Now, if we compare these sources with the meat, we can realize that all these body fluids contain very small amount, whereas meat contains very large amount per 100. Now, B12 undergoes a very curious journey in our body, and there is a role of transport proteins. These transport proteins should be understood before we undertake that journey. One is intrinsic factor or Castle's intrinsic factor, which is a glycoprotein, whereas transcobalamin 1, also known as heptocorrin, is also a glycoprotein found in saliva and stomach. Transcobalamin 2 is the main circulating transporter of the vitamin B12, and transcobalamin 3 also plays the role. Now currently transcobalamin 1 and 3 as a group are called as R factors. We will take this R factor subsequently. As I told you earlier, transcobalamin 2 is needed for transfer across body cell membranes through receptors. Now, normally we require 3 to 7 micrograms for our daily needs, and therefore an adult may take 240 microgram total, so that adequate amount is absorbed is 3 micrograms if malabsorption is there. Otherwise 3 to 7 microgram is enough and comes from our daily food, particularly non-vegetarian sources. While foodborne B12 undergoes a series of changes in GIT, and these series of changes must be understood how B12 journey actually occurs. Now B12 binds directly with hepatocorins in saliva and stomach, and some amount is also absorbed by diffusion, which is a free amount, and normally free amount is less than 1%. Sublingually given vitamin B12 bypasses intrinsic factor route, and therefore it will be easier for assimilation. It should be understood that 90% of the total body stores of vitamin D12 are found in liver. Now let us undergo the intriguing journey of vitamin B12. The dietary B12 is complexed with food proteins. Saliva secretes R binder, which goes down in the stomach along with dietary B12. Now B12 is acid labile, but the R binder B12 complex is protected from gastric acid inactivation. In addition, stomach secretes intrinsic factor, which is a glycoprotein, and intrinsic factor at present does not bind in the stomach, but it travels down in the duodenum, where in duodenum the pancreatic proteases, the protein hydrolyzing enzymes, cleave R binder from the vitamin B12. Now B12 is free and it releases B12 for combining with intrinsic factor. So it is interesting that intrinsic factor is secreted by stomach, but binding occurs in the duodenum. So about 99% of vitamin B12 is bound to intrinsic factor. Only 1% is available for simple diffusion. Now B12 intrinsic factor goes down in the ileum and is resistant to digestion by ileal juices. And when it reaches in the terminal ileum, it binds with mucosal receptors, which are currently called as cubulin or cubum receptors, which belong to LDL receptors. And therefore, through these receptors, B12 intrinsic factor goes down into the cell. So now there is an intracellular location. Inside cell, intrinsic factor is removed by lysosomal proteases, and 80% of B12 combines with transcobalamin 1 and 3, and 20% combines with transcobalamin 2. It should be understood that transcobalamin 2 is a major important circulating B12 binding carrier. There is another transporter inside cell that is called as megalin. This facilitates entry of vitamin B12 into lysosomes cells. Now transcobalamin 2 B12 complex is secreted into portal circulation, and most of it reaches into the liver and then subsequently into bone marrow cells. About 3 microgram is secreted into bile, of which about half of it is reabsorbed and half is not reabsorbed. Therefore, it undergoes smaller enterohepatic cycling also. The cellular lysosome degrades the complex to release B12 core molecule cobalamin. So now lysosomes carry the subsequent function. So cobalamin from any source, whether we take methylcobalamin, hydroxycobalamin or cyanocobalamin. It will be now cobalamin that is formed. Now there are two steps which occur for conversion of cobalamin core molecule into active coenzyme form. In the cytoplasm, methylation reaction, and in the mitochondria, adenosylation or formation of adenosine cobalamin. Now these are two very important steps with specific purposes. Now, in the bone marrow cells, methylcobalamin acts as a coenzyme to convert homocysteine to methionine. This is one. Now, which is then converted into S-adenosyl methionine. These are the intermediates which carry very important functions in the protein synthesis and DNA synthesis. The methyl group for this conversion is dominated by 5-methyl tetrahydrofolate, that is a folic acid intermediate, which becomes tetrahydrofolate by methionine synthase enzyme. It should be remembered that tetrahydrofolate is an important intermediate between folic acid and B12 pathways. Tetrahydrofolate is needed for several biochemical reactions which require one carbon group transfer. Tetrahydrofolate accepts carbon moiety during conversion of serine to glycine, and in bargain it becomes 5,10-methylene tetrahydrofolate. 5,10-methylene tetrahydrofolate donates methylene group to uridine diphosphate, and which gets converted into thymidine monophosphate. Eventually thymidine triphosphate formation occurs, and that is important for formation of DNA. Therefore, directly and indirectly, B12 is needed for DNA synthesis also. This is shown that in B12 deficiency, accumulation of 5-methyl tetrahydrofolate occurs, which cannot subserve the function further, and deficiency of tetrahydrofolate occurs, and this is also known as methyl folate trap. Is a classical methyl folate trap, that folate cannot be further utilized or metabolized in the absence of vitamin B12. Similarly, accumulation of homocysteine, methylmalonic acid, and methylmalonyl coenzyme A occur in B12 deficiency, and they can be the surrogate markers of B12 deficiency. Depletion of methionine and S-adenosyl methionine reduces formation of proteins also. What is the role of B12 in nervous system? Methylcobalamin is needed for synthesis and maintenance of myelin sheath. There are several mechanisms proposed for myelination: formation of palmitate from acetyl coenzyme A, formation of lecithin, a myelin sheath protein, formation of myelin basic protein, synthesis of myelinotropic cytokines and growth factors, and upregulation of neurotropic gene factors. And all these are reduced in the absence of vitamin B12. Apart from this, there is regulation of normal prion proteins in CNS. Adenylcobalamin dependent methylmalonyl coenzyme A mutase activity is reduced. That results in reduced generation of propionate when there is deficiency. Poor myelination is seen in vitamin B12 with deficiency of methionine synthase. Reduced transmyelination causes less availability of S-adenosyl methionine, which is required for synthesis of biogenic amine also, such as dopamine and other neurotransmitters. Phospholipids and proteins in the brain are also reduced. This may explain neuropsychiatric manifestations of vitamin B12. Adenosylation occurs mainly in mitochondria, whereas methylation occurs mainly in cytoplasm lysosome. Therefore, formation of adenylcobalamin from the core cobalamin in mitochondria occurs by enzyme adenosyl transferase. Now, adenosyl is obtained from ATP. Adenylcobalamin is a cofactor for enzyme methylmalonyl coenzyme A mutase that catalyzes conversion of MMCoA to succinyl coenzyme A. Now this is a very important step because succinyl coenzyme A is important intermediate that links carbohydrate and lipid metabolism. It is also needed for synthesis of porphyrins. And this is also important in the extraction of energy from proteins and fats through citric acid cycle when needed by the. This conversion is also important because utilization of valine, isoleucine, threonine, methionine, so amino acid utilization, cholesterol and oat chain fatty utilization can also occur through this pathway. This step is essential because it is needed for production of blood and maintenance of normal cerebral and nervous function. It is also involved in synthesis of thymidylate, necessary DNA synthesis. So all these reactions would require not only methylation but also adenine. Another intermediate form is glutathione cobalamin. Now this is another active form, less known but another active form, which acts as a cofactor in nitric oxide production and plays role in reactions which are associated with cell membrane integrity. Therefore, indirectly, vitamin B12 is needed for integrity maintenance. Supplementation with adenosylcobalamin has been shown to modulate immune system by down-regulating excess inflammatory processes which are mediated through inducible nitric oxide. So these relationships are important to understand the intricate role of vitamin B12 and its various. What are the tests for deficiency? Conventional classic was Schilling test, one step, two or three steps, which is rarely done nowadays because now there are methods available for direct estimation of levels of vitamin B12. Holotranscobalamin levels can also be measured, which also tell us about the bound form of vitamin B12 in circulation in body. Currently, commonest tests done are vitamin B12 levels. Normal levels measure free and bound form, and they vary from 300 to 900 picogram per ml. When they are less than 200 picogram, we say it is deficiency, and between 200 to 300 picogram per ml, it is depletion, and treatment must be started. Now, as I discussed earlier, elevated serum urinary methylmalonic acid is also a specific indicator of B12 deficiency as it is not affected by folic acid levels. So, serum and urine MMA levels should also be ordered when we want to know that folate is not playing the role. Elevated homocysteine is relatively more sensitive, whereas MMA is more specific. The homocysteine levels are more sensitive but not specific for as surrogate markers of B12 deficiency. Normal homocysteine levels are considered less than 15 micrograms. Now, what are the doses of vitamin B12? Replacement doses of B12 are somewhere around 1000 microgram or 1 milligram of hydroxycobalamin given weekly, and 6 doses are given. More frequent doses can also be used if there is overt neuropathy, but there is no evidence that high doses produce better results. Maintenance dose for vitamin B12 even when there is established deficiency after repletion is 1000 microgram intramuscular once in every 3 months. So we realize that in all clinical cases and physicians and prescribers are using bit higher doses of vitamin B12. Fortunately, there are no serious adverse effects with this agent. Now, because cyanocobalamin is considered to be less retained, it is given more frequently, that is 1000 microgram intramuscularly monthly. Now, if we look at the differences among these four congeners, cyanocobalamin, nitroxycobalamin, methylcobalamin, and S-adenosylcobalamin, all are available in oral dosage forms. Intramuscular dosage form of cyanocobalamin, hydroxycobalamin, and methylcobalamin are available. Intravenous form of hydroxycobalamin is available for cyanide poisoning only, and other dosage forms are being developed. For example, cyanocobalamin nasal inhalation containing 5 microgram per meter dose inhalation is also available, whereas sublingual methylcobalamin is made available, which contains 750 to 500 micrograms. Now, as I told you, doses vary, but 100 microgram daily for 7 days, then once a month is the standard dose, or 30 microgram hydroxycobalamin for 5 to 10 days, and then 100 to 200 microgram once a month, or methylcobalamin 500 to 1.5 milligram, 500 microgram to 1.5 milligram per day. Now, it is to be understood that cyanocobalamin is not used in Leber's disease because of worsening of optic atrophy. And there is a potential for cyanide poisoning because it contains about 20 microgram cyanide per 1 milligram of the drug. Although these are very minuscule amounts, but this can be also considered and kept in. Hydroxycobalamin is definitely indicated in cyanide poisoning as IV infusion 70 milligram per kg. A second dose can also be given. No other vitamin B form should be used. Now, what is practically important is there any advantage of using methylcobalamin or as adenosylcobalamin? It should be understood that all forms are equally bioavailable. Therefore, if required, any of these can be given for supplementation, replacement, and maintenance. And all forms are initially converted to cobalamin core and then converted into coenzyme form inside the cell. Therefore, exogenous methylcobalamin or adenosylcobalamin will offer no advantage because it must be converted into core molecule, that is cobalamin core molecule, that will then be converted into methylcobalamin in lysozymes or the adenylcobalamin in mitochondria. So these forms must be reconverted back to core and then to the active biological forms. Some studies show that vitamin B12 retention is longer with 3 other forms than cyanocobalamin. So less frequent doses may be required with other forms. Cyanocobalamin may be given more frequently, as I told you, once in a month. And there is also greater excretion of cyanocobalamin in urine in some individuals. Now, there could be a lower ability to convert cyanocobalamin to cobalamin core in those individuals who show genetic polymorphism of the enzymes needed for conversion and uptake of cyanocobalamin inside cells. And therefore, these various genetic variations are in the form of SNPs or single nucleotide protein changes, and therefore in these patients cyanocobalamin may not be suitable for clinical use. However, in general, all are equal. Many uses are there for vitamin B12. I will not go into detail except I will enumerate them. Pernicious anemia, megaloblastic anemia, subacute combined degeneration. In these conditions, aggressive therapy must be used. Doses I have already told. There are other diseases where there may be some role. Many studies show effects, other studies show no effects. So there are unsubstantiated claims for the treatment of diabetic neuropathy unless deficiency is there. Multiple sclerosis, chronic fatigue syndrome, myalgic encephalomyelitis, Leber's hereditary optic atrophy, where hydroxycobalamin is used, peripheral neuropathy in elderly who are on the restricted diets are not able to take the non-vegetarian food, and cervical myelopathy with neuropathic manifestations. So, apart from this, in last I will discuss the few drugs which are taken on long term can really cause vitamin B12 deficiency, and they are very important also. Today, proton pump inhibitors and metformin are extremely commonly used because of high prevalence of the gastric acidity, GERD, and peptic ulcer disease and also diabetes. Now, metformin causes B12 malabsorption by multiple mechanisms, either by affecting entrance factor B12 complex or by interfering with binding with tubulin receptors in the enterocytes, or there is reduced secretion of entrance factor, that is more important when the gastric pH is high. In the presence of high pH, the oxyntic cells do not secrete intrinsic factor and pepsin. Therefore, there is B12 malabsorption. Proton pump inhibitors also do the same because by increasing the gastric pH, they will inactivate the pepsin and also intrinsic factor secretion is affected. Alcohol and H2 receptor blockers, antacids will also reduce absorption. And about nitric oxide will cause inactivation of vitamin B12 through oxidation. Therefore, these are the four group of drugs which should be when used for long time. They are established reports, although not the carefully controlled reports or the systemic analysis, but proton pump inhibitors and metformin should be kept in mind as potential causes of B12 deficiency, particularly in those diabetics or GERD patients who are taking them for long and also are vegetarians. So in the summary points, we can say B12 is intriguing vitamin which has a critical role in cell growth, brain development, the proper nerve function and formation of red blood cells. It is needed for DNA synthesis. It is needed for cellular energy production and utilization. It reduces homocysteine, so may be cardioprotective, reducing risk of atherosclerosis. And supplementation is needed in vegetarians, elderly, and diabetics, and for the treatment of anemia with iron and folic acid.